So we left off looking at base substitution, which is a type of mutation where a single base changes. Okay, we can also get nonsense mutations, which are a type of base substitution. In this case, you change a single base. Rather than coding for a different amino acid, it changes it to a stop codon. And so if this happens, your protein is simply incomplete and it likely will not function in any way that it's intended to function. Okay, so these nonsense mutations, can be very detrimental. Whereas in our missense, these sickle cell red blood cells can function to a degree, certainly not as well as normal functioning red blood cells, but they do still work. And so they just don't work as well. But with a nonsense, you don't have a functioning protein at all. Looking at a frame shift, this happens when we have an insertion or deletion. So either an extra base is added in or a base is removed during this process of either DNA replication or transcription. And so what we see is if we get this insertion or deletion, comparing it to our model up here at the top, it's going to change every single amino acid that follows that insertion or deletion. So we aren't just changing one amino acid here, we're changing everything down the line. This can have huge impacts on the function of a protein. Okay, so not good to have a frame shift mutation. It's likely that these are going to be worse than our base substitution mutations. Okay, because base substitutions might only change one amino acid, frame shifts are guaranteed to change everything down the line. Okay. So examples of mutation and disease. Sorry about the formatting here. When it copied over, it did not do a very good job. So we've got a mutated gene code for a non-functioning protein for an enzyme. So we have some sort of mutation here. And so we've got this enzyme that no longer fits its substrate. It can't fit into the active site because its shape has changed. So we can no longer break down this molecule like it normally would have. That molecule builds up within the body and it causes some disease, potentially death. Okay. In the case of Tay-Sachs disease, we have an enzyme in our brain that breaks up fats that are formed in the brain. Tay-Sachs disease is a disease where that enzyme isn't made correctly, so they can't break down those fats, and so these fats build up in the brain, eventually causing death. Okay. So again, base substitution, we have missense, where it changes one amino acid, which doesn't create the correct protein, but it has the potential to continue working correctly to a degree, not as well as the normal one. So sickle cell anemia was that example. Androgen sensitivity is another example. This is a mutated gene, probably from some sort of frame shift. So the androgen receptor on a cell that picks up androgen, the hormone, isn't made correctly. So even though an individual is XY, these are the actual sex chromosomes that they have, which is typically associated with males. They can't recognize this androgen. Okay, so things like testosterone, these hormones. So they present as female. They do not menstruate, however, and they do have testes, but they stay in the abdominal cavity because this testosterone isn't able to be picked up on the cells to trigger other various things like secondary sex characteristics to happen in these individuals. Okay. Other things that we can have happen, you can get entire genes deleted in these processes. Genes can be moved from one area to another. We can duplicate genes. These can have varying impacts on an organism. Things that cause mutations are known as mutagens. Carcinogens are a specific class of mutagen that will cause mutations, specifically that can cause cancer, so cause the overreplication of cells. Okay, interesting. We look throughout history, more doctors smoke Camels than any other cigarette. Right before we knew that things like smoking had carcinogens in it, and they could lead to lung cancer, something like 10 to 20 times the threat of lung cancer. We had ads like this, right? And now there are still doctors that smoke, but it's probably a lot more rare than it was in the past. Okay, and so we've got these various carcinogens and mutagens here, the type of cancer that they cause, and where you find them. There are a lot of various chemicals that are bad for our DNA. Okay, so things that you will want to review here: compare and contrast the structure and function of DNA and RNA. Okay, so they differ in the sugar. DNA has deoxyribose, RNA has ribose. Their nitrogenous bases are different. DNA has thymine, while RNA has uracil. The other ones are the same. And then their structure: DNA is double-stranded, RNA is single-stranded. Okay, think about where a mutation would be most detrimental if it happened in replication, transcription, and translation, and then why you think that. And then which is going to be more severe, a point mutation or a frame shift. Okay, so which one is going to have a larger impact on how that protein is going to function at the end of these processes. Okay, and that is the end of chapter 9.